Karen Spalt from StratiCELL, and I will talk about how transcriptomic analysis perform after the New Age diet. First, a quick definition about what is transcriptomics. It's the study of the transcriptome. In other words, the complete set of RNA transcripts that are produced by the genome using high-throughput methods such as microarray analysis. The comparison of gene signatures allows for the identification of genes that are differentially expressed in distinct cell populations or in response to different treatments. For the New Age project, we couldn't study the whole genome, so we did a selection of inflammation and immune function related genes. Practically, we use a PCR Tachman low density array called Human Immune Panel from Applied Biosystem, which is containing 90 genes related to inflammation and immune function. The gene expression study has been performed from PBMC, which are promising a tissue target in the field of nutrigenomics because they seem to reflect the dietary modification at the level of gene expression. The PBMC were collected at T0 and T1 from blood of 60 patients belonging to the dietary intervention group and from 60 patients belonging to the control group, so no dietary intervention. All the samples we analyzed were coming from Italy. Here is the list of genes that has been studied, so that were in the array. As you can see, you have a lot of interleukins, some chemokine and chemokine receptor and ligands, some stress response protein, apoptosis-related genes, transcription factor, etc. You have also on the array 6 housekeeping genes, which will be helpful for the analysis. Before to do our transcriptomic analysis, we had to isolate the RNA from the PBMC we received. We did it with a commercial kit from QIAGEN. Then the RNA has been quantified and its quality has been checked by capillary electrophoresis. Those pictures shows shows at the left side intact total RNA and at the right side partially degraded RNA. You can see for the intact total RNA you have the two bands corresponding to ribosomal RNA, and when RNA is degraded you have a spread of the ribosomal bands and a high background. Among the two-fold 60 sample we received, we could only obtain good quality RNA from 41 samples at T0 T1 within the dietary intervention group, and from 45 samples within the control groups. So we did our analysis only from those samples. After RNA isolation and quality checking, we had to convert our RNA into cDNA and then to perform our PCR using the Human Immune Panel microarray. Threshold cycles were obtained for each gene, and the data were analyzed using a specific software designed to perform relative quantification of gene expression using comparative CT method. For each subject, fold change was calculated to compare each gene expression level before and after the diet intervention. Here I put the formula used to calculate the fold change, where delta CT is the CT of the gene of interest minus the CT of the housekeeping gene. Here we choose for our analysis GUSB as the best housekeeping gene for the analysis. So we collected a lot of data, and we had to do a statistical analysis from those data. We did a two-tail pair student t-test from delta CT data comparing T0 to T1 for each subject in each group. In the dietary intervention group, we could 10 genes were differentially expressed after the dietary intervention. The list is given in this table. I will go deeper in the analysis in a couple of slides. For the control group, 11 genes were also differentially expressed after the 1 year period, so it was a bit surprising because we didn't expect to have those change because no dietary intervention has been done for those patients. So now here is just a quick interpretation of the data we get. We had a look at the literature to see if we can correlate our data with what we found in the literature. So the CCR2 gene is in the dietary intervention group. The CCR2 gene coding for the chemokine receptor 2 is upregulated after dietary intervention. This gene has been shown in the literature to be a good marker of good cognitive capacity in the elderly. The two CD molecules, CD68 and CD8A, as well as the chemokine receptor 3, have been upregulated after the dietary intervention. They have different roles in T lymphocyte activation, and those increases in expression could correlate with an improved capacity to recognize microbial antigen and to an improved immune defense. The gene coding for the interferon gamma is also increased after the dietary intervention, as well as the transcription factor STAT3, which is a transcription factor activated downstream of cytokine signaling, important for the function of multiple immune cell types. It has been shown that HT regulatory cells, which are specialized CD4 T lymphocytes that suppress excessive immune response, show reduced STAT3 activation, suggesting a role of STAT3 in autoimmune regulation. And finally, the gene coding for the TNF-alpha, which is a pro-inflammatory cytokine, is also upregulated after the diet. Among the genes that are regulated in the two groups, we found 3 genes that were upregulated in the two groups, so in the dietary intervention group and in the control group. TGF-beta1 is upregulated. It's an anti-inflammatory and immunomodulatory cytokine. The gene coding for the P-selectin, which is a membrane glycoprotein that participates in mediating adhesion of leukocyte on endothelium, is also increased. This has been shown that this protein is also rapidly upregulated upon inflammatory stimuli. And the proto-oncogene SCY is also upregulated. It has been shown that this is highly expressed in T cell, and it can act as a transcriptional activator or repressor. Regarding the interpretation of the data in the control group, the CCR7 gene is downregulated. This chemokine receptor is in the literature, we found that this is often downregulated in the elderly. The interleukin 15 is also overexpressed in the control group. This interleukin is known to be abundant in blood of old people. The interleukin 4 is decreased after the 1 year period in the control group. This downregulation may reflect a decline in immunocompetence. The pro-apoptotic gene BAX is also upregulated. Its expression is increased in lymphocytes from aging subjects compared to young subjects. And regarding the guanylizine, this protein with antimicrobial activity is also, the gene coding for the protein with antimicrobial activity is also upregulated. This gene has been shown to be upregulated in T lymphocyte following IL-15 and IL-2 induction. And the next and the final gene we talk about is the perforin, which is also upregulated and which plays a role in response to several common human viral pathogens, as the cytomegalovirus and influenza virus. So that's it for our interpretation of the data comparing intervention group and control group. After that, we try to do an analysis, a similar analysis following the gender in the dietary intervention group. And we found, as you can see in the two tables below, that the genes that are differentially expressed after the dietary intervention are different in the two, so it seems to have a gender effect regarding the gene expression analysis. So, except it for STAT3 and CELL-P gene, all the genes differentially expressed as different. So that's it, basically, for our analysis for the moment. And I thank you for your attention.
Thank you very much.